Konnichiwa guys, welcome to our channel Loops of Biology. I'm Yogiraj Jakkal taking you all to the next part of biomolecules. Now, as we have studied what are the different classes of enzymes and what are the different properties of enzymes, now we have to move on to the enzyme action and its function. For their function, some of the scientists have came up with different models of enzyme functioning. So let's see why the models for enzyme functioning has been discovered. So if I'm trying to see that there is going to be enzyme interacts, enzyme interacts with with substrate. However, no one was known how the particular enzyme is fixing with the substrate or how the substrate is fixing within the enzyme. So there was unknown information information about the interactions of enzyme and substrate. So whenever the scientists wanted to think how this particular substrate is fixing inside the enzyme, then they try to model the enzyme into different format. So the fixation of substrate, the fixation of substrate with enzyme should be like a puzzle, should be like a puzzle. So whenever I'm considering a puzzle, that puzzle's block has to fix inside the cavity. Got my point? So whenever the substrate has to bind with enzyme in a site called active site of enzyme. So whenever the enzyme is present without a substrate and without some of its more structures, all right, so the inactive enzyme is called as my apoenzyme, right? I have cleared it. And whenever the enzyme is loaded with your prosthetic group, your cofactor, your coenzyme, and finally it is attached to my substrate, that complete active enzyme is known as holoenzyme. Got it my point, guys? So here there is going to be the case of apo and holo enzyme. So now it comes with what are different model of enzyme functioning has been seen. So these models were formed for understanding enzyme substrate interactions. Got it? So how substrate binds with an enzyme. That was the prime role behind designing this particular model. So there were two models. First one is called as lock and key model. And the second one is basically called as induced fit model. So there are total two models. First one is lock and key model and second one is induced fit model. If you are looking for the lock and key model, then this particular lock and key model suggests that the enzyme is basically like a lock and substrate is like a key and substrate is like a key. So whenever I'm seeing this, the enzyme is having some site for binding of the substrate. The substrate has to be of a perfect size and shape to bind inside the enzyme. Whenever the key of that particular lock binds with it, then the lock will get open. So these products are going to get formed. So if this particular model is considered, the model's analogy was first given by Emil Fischer. So the model was discovered by Emil Fischer in 1894. In 1894. When Emil Fischer saw that the enzyme should have some active site, so enzyme is going to possess active site. But this active site should be of relevant shape and size. Relevant shape and size that of substrate. So that was its hypothesis, that the enzyme active site should be according to the size and shape of the substrate. Whenever this particular site is acting as a pit inside a lock, where the key is going to be fixed, and this key is going to come up, and this key was nothing but your substrate with proper shape and size. Along with shape and size, what do you think, guys, that one more factor is important for this particular model? Can you think? Yes, that is nothing but your orientation of this particular substrate. So along with shape and size, the orientation of attachment, of attachment was also important. So in this particular case, the key, that is nothing but substrate, is fixing within a lock of the enzyme, and whenever the key is specific for the lock will get open. So whenever it is fixed inside a lock, there will be enzyme substrate complex is going to be formed. This complex will further interact with each other to form enzyme product complex. However, the attachment of product with enzyme is very, very less. That's why the product is removed out and the enzyme is isolated back. The enzyme is isolated back into the system. Got it? So what are the disadvantages of this particular model? The model has been given by the mission. It was quite interesting model that enzyme must be having some specific size and shape active site. However, there are some disadvantages of this particular model. First case, the enzyme number in biological system will increase a lot, right? Whenever the enzyme number in biological system will increase a lot, the complete space of the ground substance will be occupied by the enzyme. So it will occupy maximum space. Of ground substance. substance. But that is not the case. Got it? So in this particular case, the enzyme number will increase and they will start occupying the maximum space of the ground substance. Second case, if I try to make a specific shape enzyme, then for each substrate there will be different enzymes. So the nature of enzyme and the structure will be completely dependent on substrate. But that is not the case. This was also the flop thing. Next case, if new substrate has been arrived, then there will not be any enzyme present inside the biological system. So that will be a problematic case that our body has to get mutated and to form this particular enzyme specifically for the substrate. So there will be no enzyme. There will be no enzyme for a new substrate arriving in the biological system. So that is also one of the problem. And fourth case, sometimes one enzyme can handle many of the substrate. Sometimes one enzyme can handle many of the substrates. So there should not be two keys of the same lock. So that's why this lock and key model failed. So sometimes the enzyme can deal with more than two substrates, and where one lock cannot have two keys, right? So that's why this particular model was failed. These were the disadvantages of your lock and key model. The next model, which was came up with Koshland, the scientist Koshland gave this particular model in 1959. So this Koshland model, which was induced fate model. Now the name suggests that the substrate is inducing some changes inside the enzyme so that the substrate will get fit inside your enzyme active site. Got it? So in this particular model, whenever the substrate arises, it induces enzyme. It induces enzyme to change the shape of active site. And then it is going to form enzyme substrate complex. Then there will be the reaction take place. Then there will be enzyme product complex. And finally, enzyme plus product is given out of the system. That was a simple case of induced fate model. So unlike a lock and key, which was a rigid model, which was a restricted model for enzyme and substrate, this was a flexible model. So the model showed that the active site of enzyme that the active site of enzyme is is flexible towards towards substrate. So the active site of enzyme is flexible towards substrate. That means the active site, which is also called as substrate binding site, can change according according to the substrate in surrounding. According to the substrate in surrounding. All right. So in this particular case, how does this particular enzyme is going to change its active site? So whenever the active site has to change, the active site has to change, the bond formation in active site and substrate, alters. If suppose the enzyme is interacting with triangular substrate, and forming in suppose 3 hydrogen bonds. So this is specific number of hydrogen bonds for that particular triangular substrate. That triangular substrate is basically forming 3 hydrogen bonds along with the exposed element on that particular substrate. It's not like that every triangular substrate will form 3 hydrogen bonds. So in this particular case, the bond numbers are dependent on the exposed element for the binding. If suppose enzyme is loaded with some square shaped substrate, whenever the enzyme is loaded with my square shaped substrate and it is still forming 3 hydrogen bonds, so the specificity of enzyme is dependent on interaction of substrate with the active site. However, suppose enzyme is loaded with some square substrate and it is forming only 2 hydrogen bonds, then this substrate is not specific for that particular enzyme. So complete activity of induced fit model is basically based on how this particular substrate is interacting with enzyme inside the active site. Got my point, guys? So for this particular case, one problem has been seen that there are some inhibitors. There are some inhibitor molecules. Now there are two cases of the inhibitor. One can be reversible, one can be irreversible cases. So there are two cases for your inhibitor molecule. First case will be reversible. Now what is reversible condition? That whenever the inhibitor binds to enzyme, then The molecule can be removed out as well. The molecule can be removed out as well. It's like a family pressure. Whenever the substrate concentration is increased, whenever I'm increasing the substrate concentration, like your family pressure, in that particular case, the inhibitor will remove out easily and the enzyme will be active again. And your enzyme will be active again. Such case is of reversible inhibitor. But in some cases, it is irreversible. It is irreversible. So in case of irreversible, the enzyme inhibitor complex, the enzyme inhibitor complex is permanent, and if we want maximum enzyme to get worked, if we want maximum enzyme to be in working condition, then competitive substrate has to be added. If we loaded high amount of competitive substrate, then they will fight with this particular inhibitor to go and capture the active site first. So it is completely dependent on the volume or concentration of the substrate. Sometimes there are two more cases of inhibitor that substrate and inhibitor mimics each other. Inhibitor mimics each other. That means the shape and size of the inhibitor and substrate is same. While in some of the cases, inhibitor forms same number of hydrogen bonds as that of substrate. So how many hydrogen bonds the substrate is going to form with the active site of enzyme, the same number of hydrogen bonds are formed by the inhibitor as well. Got my point? So this was a case of inhibitor molecule. So now the question is how this particular active site is governed or changes its shape according to the substrate. Right? So there is one more site for some of the enzyme. This is my extra site on enzyme other than active site and cofactor binding site. Sometimes the enzyme has to regulate specifically and very, very carefully. Got it? So in that particular case, the cofactor is not the only or the sole element to govern the activity of active site. So in some of the cases, in some enzymes, the cofactor cannot solely govern the active site for binding of substrate So in such cases, the enzyme has one more site that is nothing but allosteric site. So the extra site that governs or regulates the changes of active site The changes of active site is nothing but my allosteric site. Is nothing but my allosteric site. In this particular case of site, whenever some molecule such as CAMP or ATP or ADP, etc., is binding on allosteric site, this site alters three-dimensional structure of protein. That is nothing but three-dimensional structure of enzyme. Now, whenever the three-dimensional structure is completely altered, that means allosteric site changes the set of exposed amino acid in the active site of enzyme, and as the set of amino acid is changed, or somehow they have switched some of the amino acid, so in that particular case the enzyme can bind to more than one substrate. So enzyme can bind to one substrate once and then to some other substrate in the next set of reactions. So the changes helps enzyme to bind to different substrate with higher affinity, where I am saving the particular energy, where due to which the total energy for new enzyme formation is saved basically. So in this particular case, I am saving up the energy, and with less energy, I am regulating more than one substrate through an activity of single enzyme. Got my point? So single enzyme is handling more than one substrate to form different product based on this particular allosteric site. So these are basically one enzyme can handle more than one substrate based on different set of reaction to form, suppose enzyme plus substrate 1 is going to give me enzyme substrate 1 complex and finally enzyme plus product 1, and enzyme plus substrate 2 is going to give me enzyme substrate 2 complex and finally going to give me enzyme plus product 2. So basically I am obtaining two different products using the same enzyme and using two different substrate over here. So allosteric site is very, very important to regulate the complete site of the active site where the substrate is going to bind or the inhibitor is going to be bind at that particular site. Got my point, guys? I hope you liked this video and understood every concept about the models of enzyme along with the allosteric site, which was new to you. And please do not forget to like, share and subscribe to our channel and share our video mostly within your friends and families. Hope you like this video. See you in the next video, guys. Bye-bye.